Okay, it's clearly impossible to talk about mechanisms of differentiation in 15 minutes, so I'll just highlight some recent exciting events in this area. And so it may sound a little bit disjointed, and I apologize in advance for that, but that's the limitation of the time. Okay, I have nothing to disclose. And so to talk about differentiation, we need to first briefly touch on the origin of these cells. And classically, we believe that there is a progenitor cell in the organ itself that survived from development that is the precursor for most of the cells that you see in tissues. And since I work mainly in pulmonary fibrosis, I have here a picture of the lung. Okay, and certainly there's support for this concept. And early on, we studied pulmonary fibrosis in a rat model, and we showed that by careful kinetic analysis that the origin is probably the peribronchial or perivascular adventitial fibroblast. On the basis of, I don't think you can see it, in situ hybridization for the collagen gene expression, and peroxidase staining for alpha-actin. But pretty soon, around the beginning of the new millennium, there are reports that there is a contribution, a possible contribution by extrapulmonary, in this case extrapulmonary sites, especially the bone marrow, and there's evidence that the hematopoietic stem cell may be a precursor cell for at least some of the structural cells in the lung. There was indeed some support by 1994. This is from Bucala's group, and they showed the presence of circulating cells that express collagen 1 and 3, CD45 and CD34, indicating its hematopoietic stem cell origin with certain properties, including the expression of collagen. And when they isolate these cells, tag them, and reinfuse them, they can find them at sites of wound healing, indicating a possible contribution from these sites. So we got interested and used a different approach and not rely on these markers, which are somewhat difficult to track, and instead use a bone marrow chimera mouse, wherein we transplanted. You can use multiple tags, but in this case we use bone marrow from transgenic mice that express green fluorescence protein, and thus label all the cells from the bone marrow. So this is a stable engraftment model, and track the presence of GFP positive cells in the lung using this bleomycin model, which, if some of you are not familiar with, consists of just endotracheal injection of bleomycin, which causes lung injury and rapid fibrosis over a 2 or 3 week period. And when we analyze the fibroblasts in the lung, we find these green cells that express collagen. But unfortunately, they do not express, do not become myofibroblasts. So you can see the alpha-actin stain do not co-localize with the GFP staining or GFP signal. In contrast, telomerase reverse transcriptase, which we found to be induced in lung fibrosis, can co-localize in some of these cells. And collagen obviously will co-localize, as shown here. And we also show that indeed these cells will not become myofibroblasts even when you stimulate them or treat them with TGFbeta, which is the classical signal for inducing differentiation. And we, just briefly to show that this is not unique to the bleomycin model, we showed this also in an airway asthma model where there is remodeling, significant remodeling of the airway wall. And so what we are left with is that this is probably an unlikely contribution, and that it's probably, I don't have time to show you the data, that there's evidence that this cell is probably functioning in a paracrine manner by releasing mediators such as TGFbeta that will then induce a local differentiation and emergence of the myofibroblast from local precursor cell. But more recently, there's been excitement with respect to the possibility of epithelial to mesenchymal transition or endothelial mesenchymal transitions. And there is precedent for this with respect to cancer as well as during development. And so early on in the studies were, I think, initiated probably in the kidney with renal fibrosis models. Eric Nielsen's group, they purport to show this. And then subsequently there was some evidence for this. But this is rather controversial. And the level of contribution by these two sources are not entirely clear. And the latest paper, which is in renal fibrosis in the unilateral ureteric obstruction model. And the latest word is not the final word, so this is certainly not final. But in this elegant study, they use lineage tracking. So this is essentially the more definitive way to look as far as the origin of cells. And they use two transcription factors that will allow them to tag either the proximal or the distal nephronic epithelium and cross them. These are driving the enzyme Cre, and cross them with mice with red fluorescence protein in this case, so that when they cross them, the Cre will activate gene expression and tag those cells at a certain point in time. So using this approach, they were able to tag these cells and then follow and see if they become myofibroblasts. And what they showed is that there is no evidence of colocalization between actin as well as that red fluorescence protein, both in the HOXB7 Cre cross mice or in the 6-2, as you can see here. There is no colocalization whatsoever. And this is just one where all the cells are tagged red, and you can see, so this is just to show that it's not the genetic makeup of these animals that's causing this kind of picture. So they concluded from this study that there is no evidence for epithelial mesenchymal transition in vivo. They were able to show it in vitro in isolated cells, but not in vivo. So what we're left with is, this is taken from a review by our chair here, and with a model in which the myofibroblasts predominantly arise from probably the perivascular adventitial fibroblast, and primarily due to stimulation by TGF-beta. And I show you a few other things here that I'll just touch on briefly in subsequent slides. In addition to inducing differentiation, the TGF-beta, we showed this a number of years ago, can also promote survival of the myofibroblasts. So in addition to inducing differentiation, they can also inhibit apoptosis in the myofibroblasts, in this example, induced by nitric oxide that's released by interleukin-1 activated fibroblasts. And thus, the TGF-beta is a potent signal for both the differentiation as well as the persistence or survival of the myofibroblasts. So it behooves us to then understand fully how this happens with respect to response to the TGF-beta. And in terms of looking at differentiation, you look at signaling pathways, gene regulation, and so on. And looking at TGF-beta, the known pathways are quite complicated. They, in addition to the I guess you can call it canonical SMAD pathways, with activation ultimately of SMAD2 and 3, which can then go into the nucleus and activate gene expression. There is also activation of the MAP kinase pathways. And subsequently, it is shown also that these pathways can interact and amplify each other, including even now the role of NOX4 and the role of oxidants in also mixing up these complicated pathways. There are a couple other pathways that I'll touch on, especially Notch later, but the question here is, looking at the marker for myofibroblast differentiation, do these pathways directly influence the alpha smooth muscle actin gene? If you analyze the promoter, in this case from the rat, previous studies have shown these very complicated situation wherein there are multiple elements that are present that are important in regulation of alpha actin gene in the smooth muscle cell. In the myofibroblast, it's somewhat different, but a few of these factors seem to play similar roles. For example, the TGFbeta hypersensitivity region and the TGFbeta control element, which are two areas that are known to be critical for TGFbeta induced alpha actin expression. But those elements really don't respond directly to TGFbeta signaling in terms of the SMAD element. So in analyzing for SMAD binding sites early on, we found two areas, what we call the SMAD binding element 1 and 2, which is upstream here. And this one is downstream to the transcription start site. And these are typified by the CAGA elements, CAGA, CAGA, and so on. So using that, to guide our work, we mutated those two binding elements. In this case, the SBE1 and this construct, SBE2. So we have a promoter construct with a luciferase reporter gene. And when we mutated those two areas, what is affected is the SBE1. So the upstream element is important, but has no impact when we mutated the downstream SBE2, suggesting to us that indeed there is direct regulation at the level of the SMAD binding element 1. And next, just to highlight a recent work in our laboratory is to look at another signal which appears to be TGF-beta independent. And this one is activated by this molecule called FIS1, which is also called resistin-like molecule alpha. And it's considered to be an adipokine initially, or primarily because it's initially discovered to be a product of adipocytes, fat cells, and potentially may be relevant even in Dupuytren, where there are fatty or fat deposits around these fascia. And this we found to be highly induced in our model in a microarray analysis of lung tissue, and which we confirmed by RT-PCR. And this molecule is a good activator of alpha-actin expression in lung fibroblasts, as shown here. And you can show that the protein level and mRNA levels. Just to briefly review Notch signaling, it essentially consists of a cell-associated ligand, either as shown here, jagged, delta, and so on are the ones. And binding to the Notch itself, which is a cell-associated receptor here. And upon binding, there is release of the intracellular domain, or ICD. And this is catalyzed by an enzyme complex called gamma secretase, which then allows this intracellular domain to migrate to the nucleus and activate gene expression by binding to CSL complex here, releasing the repressors and binding of what's called mastermind and histone acetyltransferase to activate gene expression. And indeed, we see that FIS1 does activate Notch signaling. As you can see here, jagged is activated. The intracellular domain is also increased, and a target gene to indicate activation of the Notch pathways has one. And looking at the jagged one, you can see that this is very rapidly upregulated. within a few minutes. And so the question is that relevant to alpha-actin expression? And as you can see here, there is evidence of activation of the alpha-actin in terms of protein by Western. You can see if you overexpress JAGET1 by transfecting a plasmid, you can see that there's further enhancement of what is seen beyond Fis1 alone. And here, this is the opposite, where we use siRNA to block JAGET expression, and you can see suppression of alpha-actin expression, indicating that Fis1 induction of alpha-actin expression or myofibroblast differentiation is dependent on JAGET1. And so you arrive at this picture or cartoon indicating that Fis1 somehow activates JAGET1, which then activates Notch in an adjacent cell to activate myofibroblast differentiation in this manner. So we further showed in that paper, in that study, that this is at the level of transcription. We can confirm that, and there is actual binding element for CSL in the alpha-actin promoter located very close to this CARG-A element here. So the question that, the other question that we need to address is it solely due to just induction, or is there active repression, and that there is a derepression phenomenon that's also associated with myofibroblast differentiation? So we focus over the next few slides on potential repressors of alpha-actin gene expression. And to summarize quickly preceding studies, there were 3 areas in which there is active suppression by Kruppel-like factor 4, or GKL, or gut Kruppel-like factor, at the level of competing with binding at the TCE with factors like KLF5. And we also showed that there's inhibition via the short form of CEBP-beta, which can happen at this level, and also KLF4 can inhibit binding of SMAD3 to the SMAD binding element. So there are potential sites where there is repression. However, these mechanisms are really induced mechanisms. So it's not a thing that one sees in a resting fibroblast, because KLF4 as well as CEBP-beta have to be induced by some external exogenous signal. So we went on to look for another possibility with respect to keeping cells in an undifferentiated state under homeostatic conditions. And our search serendipitously arrived at NKX2.5, which is a transcription factor that's normally associated as a marker for cardiomyocytes and is critical for cardiac development. And also, it was shown to be important in smooth muscle cells, vascular smooth muscle cell actin expression. So we were looking for regulators, and we didn't expect this effect with respect to repression in fibroblasts instead of smooth muscle cells. But in any case, we went and looked to see if there are potential binding sites in the fibroblasts as well for this transcription factor. And we found 3 possible sites, and so we mutated those sites to see what the effect is on promoter activity. And what we found is that when you mutate that NKE1 region, which is upstream, the most upstream, we see activation actually. So this was opposite to what we were expecting based on work with the vascular smooth muscle cells. So clearly it's behaving differently in the fibroblast. And it's clearly a repressor in this instance. And to make a long story short, it seems to be repressing differentiation. And if we suppress or inhibit NKX2.5, this is sufficient to promote differentiation. You can see alpha-actin expression, and basic FGF suppresses this via NKX2.5. And in vivo, we demonstrated that fibrosis is associated with reduction in NKX2.5 expression. So suggesting that this is a possibility with respect to keeping cells quiet and not becoming a myofibroblast under normal conditions. So presumably, under conditions of fibrosis, this gets repressed and then get activated in terms of expression. So we end up with this more complete picture wherein NKX2.5 behave as a repressor. And there is support for this because you can look at truncated, serially truncated promoters, and you find that as you shorten them, you lose inhibition as suggested here. So now I just want to end by touching on epigenetic regulation, which has become exciting also. And basically you can divide this into two areas, two major areas. One is histone modification, either acetylation or methylation, which is associated with keeping the chromosomes in a configuration that prevents gene expression, or its modification then allows genes to be expressed. And there is some support for this, and I won't show you any data from studies using trichostatin A, for example, which is an inhibitor of the histone deacetylase enzymes, and that is shown to inhibit differentiation. So, which is somewhat counterintuitive because usually the acetylation will block gene expression. So by blocking, you should activate gene expression. So this suggests that this is an indirect mechanism, and subsequent study shows that it is possibly working through other molecules, and that this is probably working through another molecule that inhibits or suppresses myofibroblast regulation, differentiation. I want to touch a little bit more on DNA methylation, which is also another mechanism for suppressing genes from being expressed, and occurs mainly in CPG islands, but recently is shown to also, it can happen in non-CPG islands, so in CA sequences and so on. And these are catalyzed by a number of enzymes called the DNA methyltransferases. They're 1, 3A, and 3B. And usually will inhibit gene suppression. We found in the alpha smooth muscle actin gene, this is from rat lung fibroblasts, that there are 3 major CPG islands that are methylated. And what is interesting about this is that when we compare the level of methylation between a cell that can express with the potential to express alpha actin, such as the fibroblast, versus alveolar epithelial cells which do not express alpha actin, we see a markedly different methylation patterns in which, unfortunately I don't know if you can see the color clearly, but the brown is the fibroblast and the greenish yellow is the type 2 cell or alveolar epithelial cells. You can see that they're almost equally methylated in the upstream CPG islands. But downstream in the intronic regions, the red lung fibroblast is very lowly or poorly methylated, in contrast to epithelial cells, which is highly methylated, which suggests to us that methylation is a differential indicator of potential for myofibroblast differentiation, and potentially also an argument against EMT being a key mechanism for myofibroblast differentiation. Anyway, looking at the methylation of the fibroblasts, we can reduce methylation by treating cells with an inhibitor called the oxycytidine here, 5-AZA deoxycytidine. And what we can see is that there's a reduction in the methylation by treatment of this agent, and we can see a dose-dependent increase in the level of alpha-actin expression when we suppress DNA methylation. And more specifically, with respect to the protein, this is mRNA, you can see effects at 5 and 10 micromolar, and the effect is more dramatic at 5 days of treatment, indicating, because this inhibitor basically will inhibit and The level of methylation obviously doesn't change, it's relatively stable, and it only changes with cell division. And we can prove this also by using more specific way of modulating this using siRNA for DNMTs. And as you can see here, siRNA to the various DNMTs can induce alpha-actin expression, confirming that methylation is an important mechanism by which these cells are being kept undifferentiated, so to speak. So quickly near the end then, I just want to cite similar studies in the liver fibrosis model using carbon tetrachloride. This is by the group of Derek Mann in the UK, and they showed that a similar story, but they attribute this to this MECP2, which is right here, which is a methyl binding protein that's associated with repression of gene expression. But they attribute their findings because what they found was really an effect more on NF-kappa B by blocking the expression of I-kappa B. And as you know, NF-kappa B has suppressive influences on alpha-actin. So the mechanism that they're suggesting here is that the MECP2 working with methylated DNA to suppress I-kappa B. But in that study, they were not able to actually show CPG islands in the promoter, nor were they able to show actually methylated DNA. So subsequently, they were able to show that this is probably working also through, or more relevantly, through PPAR gamma, which is another suppressor of alpha-actin expression. And in this study, they were showed that regulation of MECP2 through microRNA 132, which has MECP2 as a target gene. So during differentiation, there's repression of MI or microRNA 132, resulting in expression of MECP2 that went on to suppress expression of PPAR gamma and thus allow expression of alpha-actin. So there are multiple mechanisms involved here, both direct and indirect. And I just want to conclude by summarizing that these are very complex, both involving transcriptional and epigenetic mechanisms, epigenetic including microRNA these days, and that it's stimulus specific and involves multiple signaling pathways, which makes our tasks when we think of treatment more complicated. And that may be why also single treatments, unimodal treatment to target TGFbeta signaling, for example, may not work. And that there's active repression, and maybe release from repression may be an important pathway, and that there are indirect mechanisms as well. And finally, I want to thank the folks in my lab that did the work that I reported to you based on my laboratory's work, and collaborators and support. Thank you very much.